Hello, my name is Marissa Craig, and this research project was conducted on gross primary productivity comparison between evergreen and deciduous magnolia trees. It was decided to study the differences in metabolic rate between the evergreen magnolia, Magnolia grandiflora, and the deciduous magnolia, Magnolia acuminata, which is located on NKU's campus. The Magnolia grandiflora, also known as the evergreen magnolia and commonly known as the southern magnolia, are native to the U.S. As shown in their picture, their growing zones are indicated 6 through 10. Its leaves are large, dark green, and oval shaped that are covered in a thick, waxy layer with a fuzzy brown underside. If not properly trimmed, the tree will sprout multiple new branches among the stem, which gives a tall, symmetrical bush appearance. This tree is typically a representation of the southern United States and is the Mississippi state flower. The southern magnolias do not produce nectar. Instead, they produce large white flowers that have sugary scent, which attracts pollinators. The southern magnolia also produces cone like brownish fruit that can reach 2 to 10 inches in length. The Magnolia acuminata, also known as the deciduous magnolia, is commonly known as the cucumber tree. They are also native to the U.S., as shown in the picture, and their growing zones are indicated 4 through 8. Its leaves are smaller in comparison to the southern magnolia, ranging from 6 to 10 inches long, and are light green in the summer and yellowish brown in the fall. The cucumber tree can attain a height of 60 to 80 feet with a span of 35 to 60 feet and can stand a variety of temperatures. In the winter months, the trees produce a fruit, which are cones with bright red berries that provide sustenance for wildlife. Early Americans flavored whiskey with the bitter berries and were also used for medicinal purposes. Through photorespiration, plants are able to draw sunlight, water, and carbon dioxide from their environment to then synthesize glucose and other byproducts such as oxygen. The net primary productivity rate refers to the measurement of energy used by the plant while performing photosynthesis, which can then be measured with the exaspiration of carbon dioxide. Respirations refer to the carbon dioxide loss due to energy used during metabolic activity. Subtracting these two data points will result with the gross primary productivity rate, which refers to the excess energy left over after photosynthesis. This is referring to the formula of cellular respiration, which is NPP minus R equals GPP. This information could be applied to the agricultural field and then be used to help resolve humanitarian problems, such as world hunger, climate change, and help the economy. The results could be used to create genetically modified hybrids based on the collection from the deciduous Magnolia acuminata and the evergreen Magnolia grandiflora. This hybrid could be used in different species of plants, such as produce producing plants and high yield oxygen producing plants. These hybrids would contain the fast photosynthesis rate discovered in the Magnolia acuminata and the continuous photosynthesis process that occurs in the Magnolia grandiflora. For produce producing plants, this would create a major change in the agricultural field. This hybrid concept could potentially create higher crop yields with more nutrients and longer growing seasons. This factor alone could help be used with world hunger and help grow the economy. Higher crop yields with more nutrients and longer growing seasons allow for more produce to be delivered, which creates more availability to those in need. This also makes the agricultural field more profitable for those who work in it, while possibly creating new export opportunities, ultimately helping the economy. Regarding a hybrid of high yield oxygen producing plants, this could help fight climate change and deforestation. Further research has found that within the Magnolia family, a particular magnolia is a hybrid of both deciduous and evergreen aspects. The big leaf magnolia, Magnolia macrophylla, is a deciduous tree but is also semi evergreen, commonly found in the southeastern hemisphere. Hybrid species have also been made from the Magnolia cuminata, the cucumber tree, and other Magnolia species. These include the Magnolia leaflora, Magnolia springeria, and the Magnolia X southern gala. It remains to be seen how important these hybrid species have become. The most used hybrid in landscaping are the Magnolia acuminata and the Magnolia deodata. These are crosses that involve the smaller, shrubby, winter hardy, large yellow flowered magnolias. Based on Sherby's reference data, we knew that the deciduous species would photosynthesize more efficiently because they do not need to photosynthesize to create energy because it's already stored. Skipping this step allows the deciduous trees to photosynthesize quicker and more efficiently. Referencing this information allowed us to create an accurate hypothesis. Deciduous species need to harvest more energy through photosynthesis in a shorter amount of time and have higher rates of GPP than evergreen plants. Our hypothesis states that the deciduous species need to harvest more energy through photosynthesis in a shorter amount of time to ensure a higher rate of GPP than evergreen species because they go through dormancy in the winter months. Our experimental design consists of collecting a total of 12 samples. There will be a total of 6 deciduous Magnolia cuminata samples, 3 from behind the old Honors House and 3 from the grounds between the Lucas Administrative Center and the University Center. Another 6 total Evergreen Magnolia grandiflora samples will be collected. 3 from the walkway leading to the Science Center from the first floor, and 3 from the steps besides the soda building. To begin collecting data, we collected and marked 3 sample leaves and weighed them in grams while also measuring environmental factors in its natural environment, such as temperature and humidity. Next, we acclimated the leaves to its environments for 10 minutes. Then we placed the veneer CO2 probes into the research chamber and recorded the metabolic rate of photosynthesis for 600 seconds to collect the net primary productivity rate. Next, we acclimated the leaves to darkness for 15 minutes in a light blocking chamber. Then placed the veneer CO2 probes into the light blocking research chamber and recorded the metabolic rate of photosynthesis for 600 seconds to collect the respiration rate. This process of data collection will be repeated for each sample set. With this data secured, we began to calculate the gross primary productivity rate of each leaf using the formula for cellular respiration, which is NPP minus R equals GPP. To ensure no sampling biases occurred in our data pool, we consisted of different locations and different environmental factors. The control group for this experiment will be the 2 leaves taken from both a deciduous magnolia tree and an evergreen magnolia tree that were not used in the experiment. Our independent variable can be identified as the genus species of the magnolia tree, the evergreen magnolia, Magnolia grandiflora, and the deciduous magnolia, Magnolia cuminata. Our dependent variable can be identified as the photosynthesis rate produced by each sample leaf. The graphs above represent the data collected from the experiment. The 2 samples removed from the Magnolia grandiflora dataset were both positive. The 2 samples removed from the Magnolia cuminata dataset were both negative. The data needed to result in a positive GPP and a negative MPP were removed to correspond with the formula to produce accurate data. The p-value of our experiment was 0.038, which led us to support our decision to reject the null hypothesis because it was less than 0.5. Our data shows that the evergreen magnolia trees photosynthesized at a faster rate. The results support that we should refuse to reject our alternative hypothesis. This means that we support the alternative hypothesis. This suggests that there is a difference between the deciduous magnolia, Magnolia cuminata, and the evergreen magnolia, Magnolia grandiflora. I also reject the experimental hypothesis stating that the deciduous magnolia would photosynthesize more efficiently, as this is not expressed in the data. The standard deviations were large, which suggests that this data is not reliable. There are many potential reasons as to why the data is so skewed, including the plants' proximity to each other on campus, not running all the data sets in the same day, running the data collections in different locations, and considering the current seasons. This factor alone affects the data significantly, considering the deciduous trees photosynthesize at a slower rate due to dormancy. Because of the research being conducted so late in the season, the chloroplasts had already started to break down. This experiment would conclude better results if done in a warmer season, such as late spring or early summer. I strongly believe that if we had conducted this experiment in a different growing season, our data would have supported that of our reference data and would have more reflected our original prediction. Here are the cited literature and visual aids in CSE format.